This video is going to look at some of the nutritional factors that affect bacterial growth, and then we'll look to see what happens when bacteria run out of the nutritional factors that they require, how they respond in those situations. So, what are the nutrients that are required by microbes? Well, this isn't a complete list, but it gets you at least a large-scale picture of what it is they need. They need a carbon source, primarily because carbon sources provide energy, but they are also important for building other molecules. They need nitrogen and sulfur, phosphorus, and then a lot of the trace elements. Some of the metals that we see on the periodic table. And then last of all, vitamins need cofactors for the enzymes, the vitamins. Now some microbes are what is termed fastidious. A fastidious microbe is one that has very specific nutritional needs and are frequently difficult to meet in the lab. So there are some bacteria that are, because they are so fastidious, they are very difficult to grow in the lab. So fastidious is just about having very, very particular and difficult to supply nutritional needs. And here is a blood agar that it's called New York City agar, and it is a specialized agar that is that meets the needs of the fastidious bacteria Neisseria gonorrhea, the one that is responsible for the disease that we talk about as gonorrhea. Okay, so nutritional factors. Let's get started by looking at carbon. So carbon is found in all of the organic molecules, and they serve as an energy source for most bacteria, as well as the building blocks for other materials that are needed in the cell. So we use carbon-containing monomers to build the polymers that are important to cells. So for instance, we must have, or bacteria must have amino acids in order to build proteins. They need saccharides in order to build the sugars and the polysaccharides that are required. They require the fatty acids in order to build the lipids they need. They require sterols like cholesterol in order to build those kinds of lipids, the steroid lipids. And then finally, to make DNA, they require nucleic acids. So many of these they can construct on their own. They can take other materials and build them into them. But some of these monomers are required because the bacteria are incapable of making them. Finally, photosynthetic microbes, the ones that are what we term photoautotrophic. If an organism is autotrophic, that means it uses either photosynthesis or chemosynthesis to produce glucose from carbon dioxide and water. So photosynthetic microbes, they can produce glucose on their own. Non-photosynthetic microbes must be able to find a source of energy that they can then convert to glucose if they can't find glucose itself. So, what about nitrogen? Nitrogen is an important component of amino acids and the proteins that amino acids are used to build. And as we have talked about, these proteins serve as enzymes. They serve as the membrane proteins that are moving things across membranes and serving other membrane functions. And there are more, lots and lots of ways that proteins are important to bacteria and other microbes. Nitrogen is also a component of the nucleotides that are used to build RNA and DNA. So here we see a nucleotide, and notice that here in the base there's a lot of nitrogen in it. So a nucleotide is composed of a phosphate group, a 5-carbon sugar, either ribose or deoxyribose, and then this nitrogenous base. And in this base we see 1, 2, 3, 4, 5 nitrogens in it. And so in order to make the DNA or RNA that this is a subunit of, we've got to have lots of nitrogen to do that. As well, This is the generalized view of an amino acid. An amino acid is a central carbon that has this carboxyl group on one side and an amino group on the other side. The amino group is built around a nitrogen. So we can't build either DNA, RNA, or proteins without nitrogen available. Some bacteria, as I said, can synthesize all 20 of the amino acids, but others of them must have at least a subset of those 20 amino acids supplied to them by the environment. And there are even some fastidious bacteria that make no amino acids on their own. They have to have all 20 of them supplied in order to make the variety of proteins that are found in their cells. Finally, some microbes are actually able to synthesize organic forms of nitrogen from inorganic nitrogen gas. We refer to these microbes as the nitrogen fixing bacteria. And they are very important for plants. So important, in fact, that plants will allow them to make a home in their roots, in little nodules on their roots, where the nitrogen-fixing bacteria will grab up gaseous nitrogen and convert it into an organic form that the plants then can make use of to build their own nucleotides and amino acids. Okay, so that's nitrogen and its importance. How about sulfur and phosphorus? Sulfur is super important because it is found in cysteine, which is an amino acid that is found in a lot of proteins. And cysteine contains a sulfur molecule. So any protein that requires the amino acid cysteine is an example of one that requires sulfur. And we get sulfur, or bacteria get sulfur, from inorganic sulfate salts and from amino acids that contain sulfur. So we can break down other amino acids than cysteine, use the sulfur to build cysteine. So there are, I forget how many amino acids have sulfur in them. Cysteine I know of, it seems like there's one more. So that's sulfur. So it's found in inorganic sulfate salts that are in the media that we provide and are found in soils and other locations for free-living bacteria. Phosphorus is important because you can't build ATP without 3 phosphate groups. There's 1, 2, and 3 phosphate groups. So anytime we're going to build ATP, we have to have phosphorus. Anytime we're building a cell membrane, we have to have phosphorus because it's found in the polar head of a phospholipid. And then nucleic acids also contain phosphorus. So that's part of the way that we link together individual nucleotides to build nucleic acids. And all of these are obtained from inorganic phosphate ions that are found again in soils and placed into the media that the bacteria are going to grow in. Okay, so that's sulfur and phosphorus. Not last, but second to last are the trace elements. These are minerals that are found in the environment as ions, and they're generally just required in very tiny amounts, but because they're so important to the processes that they're involved in, if some of these trace elements are missing, then growth can be really limited for the bacteria. Sodium and chloride are both examples of trace elements. Potassium, zinc, magnesium, manganese. They are important because they serve as enzyme cofactors, helping to activate enzymes, making the enzymes functional. Cobalt is important because it's used to synthesize the vitamin B12. We'll talk about vitamins in a minute and their role. Iron is important as a part of the heme molecule, which is used in several of the energy-producing molecules that we find in bacteria. Calcium is important because it's involved in cell wall synthesis for gram-positive bacteria. It helps to cross-link the cell walls in those gram-positive bacteria. And a couple of other important trace elements, copper and zinc, are also important. And we're not going to get real concerned about these functions, but I wanted you to see that the variety of things that act as trace elements in the nutrition of bacteria and other microbes. The last of the nutritional factors affecting growth are vitamins. Vitamins are organic molecules that act as enzyme cofactors. So just like the enzyme cofactors we were talking about in the trace element enzyme cofactors, these, though, rather than being inorganic forms, these are organic forms of molecules that act as enzyme cofactors. And the enzymes won't function unless they have these cofactors bound as part of them activating the enzyme. They're required from the environment if the bacteria can't synthesize the vitamin. And so if you're going to build a medium for a bacteria that has specific vitamin needs because it can't make those vitamins, then you have to be sure to include in that medium the vitamins that are important to the bacterium. So some of these vitamins are folic acid, vitamin B12, and vitamin K. And it turns out that human pathogens can be difficult to grow in the lab unless the culture medium includes the vitamins that would be normally obtained from the human hosts that these pathogens grow in. And then you may have learned this already in like an anatomy and physiology course, but the intestinal microbes that live in humans are very important because they synthesize vitamin K in the gut. E. coli is a big source of vitamin K to humans, and some of the B vitamins as well. So they are of benefit, these vitamins, to the microbe themselves, and because they make so much, they're of benefit to the human hosts too. So here we see a benefit, one more benefit from having a rich and varied intestinal flora that can supply vitamin K and some of the B vitamins. So last two slides here. First is to talk about nutritional complexity. So an organism's nutritional complexity includes, or it's described by listing all of the nutrients that the bacteria has to obtain from the environment in order to grow. And this list of nutrients that must be found in the environment is determined by the enzymes that the bacteria doesn't have. And if you don't have specific enzymes, then you're not going to have specific nutrients that are required because you can't build them yourself. So the fewer enzymes that bacteria have, then the more complex their nutritional requirements are going to be. Okay, so that's an organism's nutritional complexity. Fewer enzymes mean a more complex nutritive environment required. Where do we find these enzymes that catalyze the metabolic reactions? There's two locations. They can be endoenzymes. Those are the ones that are found within the cell, and so they'll just be dissolved in the cytoplasm of the cell. And then there are exoenzymes. These are the enzymes found outside of the cell, and they're used to digest molecules that the bacteria encounters so that the bacteria can then absorb those molecules. So without exoenzymes, the bacteria cannot bring the molecules that are broken down into the cell. If they're extracellular, and we find these in gram-positive rods, they're found in the medium just outside of the bacteria's cell wall. So they're in the medium right around the bacteria. If they're periplasmic enzymes, these are the ones that are found in gram-negative bacteria that have that periplasmic space in between the cell membrane and the cell wall. So periplasmic membranes or enzymes are found in the periplasmic space, while extracellular enzymes are all the way outside the bacteria's cell wall. Okay, last thing to talk about is how do bacteria deal with the fact that nutrients will, over time, that nutrients around them in their environment, in their culture media, in the agar that they're growing on, those nutrients will be used up. So when microbes encounter limited nutrients, then they have a couple of behaviors that they will engage in. The first is that they will synthesize additional enzymes. And the goal of this is to be able to take up the few nutrients that are still available as efficiently as possible. So making use of the fact that when things are limited, then you've got to build more finders to go find the nutrients that are available. The second is that bacteria will shift their metabolic processes so that they can use alternative nutrients in place of the preferred nutrient. So as we lose the preferred nutrient, as it becomes less and less common, then they just shift to using alternative nutrients. And then finally, as a last-ditch effort to kind of survive the tough times that are ahead, they can slow their metabolic processes down. They can slow them to a rate that will allow the organism to survive, but prevents any growth, any cell division, so that they can kind of temper their metabolic processes to match the conditions, with the hope that if they can survive, if they can slow metabolism down for just long enough to survive the bad times, that perhaps environmental conditions might improve. So 3 strategies that bacteria use to adapt to limited nutrients. First, going out and grabbing more of the nutrients by making more enzymes that can then make use of whatever nutrients the organism can find. Shifting to second choice nutrients. And third, slowing everything way down in the hope of being able to survive the bad times. All right, there it is. You know how to reach me if you need me.